This is DNA, deoxyribonucleic acid. During this video, we will get different representation of the DNA molecule to better view certain details, but all views represent the same structure. Inside the cell, you will most commonly find double-stranded DNA in which two strands intertwine to form a double helix. The most common form of the DNA double helix, which is all we will discuss here, is called B-form DNA. Now let's move to a more simplified structure of DNA to discuss the details. We can unwind the double helix like this so that we can see the chemical structure inside. Each strand is a polynucleotide, meaning each strand is made up of many individual units called nucleotides. Nucleotide has three components: the 5-carbon sugar, a phosphate group, and 4 possible nitrogenous bases: adenine, guanine, thymine, cytosine. The nitrogenous base is always attached to the 1-degree carbon of the sugar. If we count from there, we can see that there is a phosphate between the 5' carbon of one sugar and 3' carbon of the neighboring sugar. The sugar is called deoxyribose because it is missing a hydroxyl group at the 2' carbon, which is present in ribose. Because of this, nucleotides in deoxyribonucleic acid are called deoxynucleotide. Nucleotides attach to each other at the DNA strand by phosphodiester bond. The phosphate group of one nucleotide binds to the 3' oxygen of neighboring nucleotide. Thus, we can see that the sugars and phosphate groups make up the DNA backbone. The carbon numbering is key to describing the directionality of the DNA strand: 5' to 3'. Look within the sugars. There is an intrinsic orientation difference between the two strands. On top strand, you can see that the 5' carbon of each sugar is on the left and the 3' carbon is on the right. The opposite is true for the bottom strand. Reading left to right, that makes the top strand orientation 5' to 3' and the bottom strand orientation 3' to 5'. These strands are also sometimes called Watson and Crick. Although the nucleotides come together through covalent bonds in the backbone, the two DNA strands interact through non-covalent hydrogen bonds between the bases. Each base forms multiple hydrogen bonds with its complementary base on the opposite strand. Bound together by hydrogen bonds, each unit is called a base pair. The hydrogen bonding contributes the specificity of base pairing. Thymine preferentially pairs with adenine through the hydrogen bonds and cytosine preferentially bonds with guanine through 3 hydrogen bonds. Thymine and cytosine are called pyrimidines by their single ring structure, and adenine and guanine are called purines, which have double rings. The geometry of the AT or TA and GC or CG base pair is the same, allowing for symmetry and base stacking in the helix. This mostly has to do with the distance between the backbones and the angles to which the bases attach to the backbone. Other base pairs like GT, for example, do not have the same geometry, cannot form strong hydrogen bonds, and disturb the helix. The double helix structure of DNA is highly regular. Each turn of the helix measures approximately 10 base pairs. In addition to the hydrogen bonding between the bases, the stacking of the bases also stabilizes the double helix structure. These pi-pi interactions form when the aromatic rings of the bases stack next to each other and share electron probabilities. The regularity of the helical structure form two repeating and alternating spaces called the major and minor grooves. The grooves act as base pair recognition and binding site for protein. The major groove contains base pair specific information while the minor groove is largely base pair non-specific. This is because of the patterns of hydrogen bond acceptors and donors that proteins can interact within the grooves. In this way, DNA can be acted upon in either specific or non-sequence specific manner, allowing proteins to position themselves correctly and carry out their designated task. This is the DNA double helix structure.